today's podcast is on the cell cycle. This is basically the life cycle of a cell. And there's a few different phases that a cell goes through. Okay, the cell spends most of its life in a phase called interphase. It was called interphase because it seemed like intermediary, like nothing much was happening. But as scientists have studied the cell, they found out that that's actually when the cell does most of the things that it does. And so there's different phases. There's G1. G stands for growth. Okay, then there's the S phase, which stands for synthesis, where DNA replicates, and then G2, where it grows more and prepares for mitosis, and then we have mitosis followed by cytokinesis. So that's what we're going to be looking at today. What are these different phases? Okay, so this is a little animation going through the cell cycle. Okay, so as you can see, okay, the cell starts off here in its G1 phase where it's going to grow. That's the DNA unzipping and copying itself so that the cell can go through mitosis. It goes kind of fast. So again, I'll slow it down, okay, and we'll go through step by step. Okay, so here is a cell that just got formed when those cells split into two. So now there's two new cells. They're going to start off in G1 where they're going to grow. Okay, and sometimes they go into this phase over here which says G0. That doesn't say go, it says G0, where the cells just rest. It doesn't mean that they're actually doing nothing, not like taking a nap resting, just they're not dividing. So a lot of times, say in our brain, our cells aren't dividing a lot in our brain, so they're probably in this G0 state. Okay, then when the cell is ready, okay, it's going to go through the S phase, where it's going to unzip its DNA, make a copy, G2, followed by mitosis. Okay, so those are the phases of the cell cycle. So G1, S, G2, and then mitosis there where you see the cell actually divide. Okay, so what happens in all these phases? Okay, let's write it down. In G1 is where the cells are going to be growing and carrying out their routine functions. Okay, so if it's a liver cell, it's going to be detoxifying poisons. If it's a stomach cell, it's going to be making enzymes to break down your food. So normal routine, whatever the cells do. Synthesis phase, okay, this is really important, where the DNA is replicated. You can just write that down. The second one just says that the chromatids are attached together. So if you just look at this picture here, this is a chromosome, and in the middle of a chromosome, which looks like an X, there's a part called a centromere. Okay, because what you're actually looking at when you see a chromosome is the two copies of the DNA. So the DNA replicates, and then over this side, that's one of the copies of the DNA. And then over here is the other copy of the DNA. And they're held together in the middle so that we make sure that we pass on one half to one cell and one half to the other cell. Okay. G2, the cell's going to grow even bigger, make more organelles so that each cell can have some when it divides. Okay, and then we go through mitosis. Okay, now why would a cell decide to go from G1 or G0 to the S phase? What makes a cell decide that it needs to divide? Okay, and there's really two reasons. One, it just wants to grow, or two, it needs to repair or replace damaged or dead cells. Okay, so what's mitosis? Well, that happens after G2. Mitosis is very technical. It's actually the division of the nucleus itself. So not even the whole cell, just the nucleus. Okay, and we'll get to cytokinesis later. Mitosis has four phases: prophase, metaphase, anaphase, telophase. Okay, this is again where you might make up something to help you remember. PMAT, PMAT, okay, or PMAT, pass me a taco, players make a team. You can use any, you can make up your own, okay, but you do need to know the four phases in order, okay, and you got to know what they kind of look like. So the first phase is called prophase, okay, and this is just going to show you the whole thing again, running through the whole thing, okay. So starting at the beginning, at interphase, Okay, we're going to go through prophase, and then metaphase, anaphase, telophase, and then cytokinesis and interphase. So watch it happen. Okay, we finished mitosis. So in prophase, okay, we see the DNA starting to form these chromosomes. We see this stringy stuff forming. That's called spindle fibers. Prometaphase, you don't really need to know, but again, it just means between prophase and metaphase. Here we are in metaphase. The chromosomes line up in the middle. Anaphase, they get pulled apart. And telophase, they get form 2 new nuclei, and cytokinesis, we form 2 new cells. And back to interphase. Okay, so again, what happens during prophase? The DNA coils up into chromosomes, so you need to write these 3 things down next to your picture of prophase. Centrioles help form spindle fibers. Okay, so see this stringy stuff up the top? Those are called the spindle fibers. The centrioles are the things that look like little mini marshmallows. And then the nucleus actually dissolves, like the nucleus disappears. It goes away during this whole process. Okay, metaphase. This is an easy one to remember because the chromosomes line up in the middle. So middle for metaphase, or sometimes they call it the equator, and the spindle fibers attached to the chromosomes. Can you guess what the spindle fibers are going to do? Okay, it's the spindle fibers that are actually going to pull the chromosomes apart. So remember, when I showed you a chromosome, one half was the one copy and the other half was the other copy of the DNA. So now we're pulling the 2 copies apart. Alright, so that's going to make sure that one copy goes to one cell and one copy goes to the other cell. And then telophase is really the opposite of prophase. Okay, everything that happened in prophase, go back, flip your paper over, look, it's just going to be the opposite. Okay, so the nuclear membrane reforms, the chromosomes uncoil, and the spindle fibers dissolve. And then last but not least, after mitosis, so technically, go back for a second, we have 2 new nuclei right here, and so technically mitosis is over. But look at the picture. Are there 2 new cells yet? Not yet. Okay, so cytokinesis happens afterwards, in which the cell or the cytoplasm actually splits itself. So here in the animal cell, the cytoplasm has to pinch together. In a plant cell, they can't move to split apart, so they just form this thing called a cell plate. So right here, okay, or right here, you can see it in the picture, it basically is going to become the new cell wall that forms between them. Okay, so there's cytokinesis happening in the animal cell. The cytoplasm has to pinch and break apart. Here it is again. So these are real life pictures, which if we want to next class, you will get to look at them under the microscope and see the onion in different phases of mitosis. Okay, but prophase is a normal old cell with a nucleus. Okay, prophase, you start to see the chromosomes coil up. Metaphase, they line up in the middle. Anaphase, they're pulled apart. And telophase, 2 new nuclei with the cell plate in the middle. And I'm sure everyone in here has heard of cancer and probably knows someone who's had cancer. What is cancer? It's basically, they call it a disease of the cell cycle. There's some chemicals, one's called cyclin, which sounds just like it is. Its job is to regulate the cell cycle, to make sure that your cells divide properly. Okay, well, cancer is basically cells dividing too much. They start dividing uncontrollably. They go through this cell cycle too much. And their regulators that are supposed to limit them don't do that. And so that's what happens. You get too many cells for cancer. So, and they're just your own cells, which is what makes them hard to kill, because they're your cells. And so it's hard to kill just the bad cancer cells without killing all the rest of your cells. Okay, so that's it for the cell cycle.